Hello and welcome to Biology Explained. Today, we are going to explore a concept that is absolutely fundamental to understanding life itself: the structure and function of prokaryotic and eukaryotic cells. Think of cells as the tiny building blocks of life. Every plant, animal, fungus, and bacterium starts as a cell. Cells are not just building blocks, they are the little factories performing all the processes necessary to sustain life. But here's the exciting part: not all cells are created equal. Some are simple and compact, while others are complex and highly organized. Understanding these differences is key, not just for biology exams, but for real world applications in medicine, biotechnology, and environmental sciences. For example, did you know that knowing the difference between a bacterial cell and a human cell helps scientists design antibiotics that kill bacteria without harming humans? By the end of this video, you will know exactly what makes prokaryotic and eukaryotic cells unique, how their structure links to their functions, and why they are central to life as we know it. Let's start with some definitions. A cell is the smallest unit of life capable of carrying out all life's processes. This includes metabolism, growth, reproduction, and responding to the environment. You can think of it as the fundamental unit of organization for all living organisms. Cells fall into two broad categories: prokaryotic and the eukaryotic. The prokaryotic cells are the simpler of the two. They have no nucleus and no membrane bound organelles. Their DNA is usually a single circular molecule floating in the cytoplasm. Prokaryotes are often single celled organisms like bacteria and archaea. Despite being simple, prokaryotic cells are incredibly efficient. They can reproduce rapidly, adapt to extreme environments, and carry out all necessary life functions without compartmentalization. Eukaryotic cells are more complex. They have a nucleus where the DNA is stored and multiple organelles. Each perform specialized tasks. Examples include plants and animal cells, fungi, and protists. These cells are larger and structurally more sophisticated, allowing them to perform more complex processes. Think of prokaryotic cells as tiny studios where everything happens in one space, while eukaryotic cells are more like a modern house with specialized room for cooking, sleeping, and working. Let's visualize this. Here's a prokaryotic cell: a single compartment with DNA floating free, ribosomes making proteins, and a simple cell membrane controlling what enters and what exits. Now here's a eukaryotic cell: DNA safely enclosed in a nucleus, mitochondria producing energy, and endoplasmic reticulum and Golgi apparatus processing proteins, and lysosomes cleaning up wastes. So real-world examples of the prokaryote is the E. coli found in your guts, the cyanobacteria, which produces oxygen through photosynthesis. While real-world examples of the eukaryote, your chick cells, your plant leaf cells, yeast used in bread and beer, and cells of simple animals like the amoeba. Now let's dive deeper into the structural differences between these two cell types. We'll go slowly and really explore each feature. Let's start with the cell wall. Prokaryotic cells often have a rigid cell wall made of peptidoglycan. This wall gives them shape, protects them against bursting, and allows survival in harsh environments. For example, bacteria can survive acidic stomach conditions or hot springs because of this sturdy wall. In contrast, eukaryotic cells only have cell walls in plants, fungi, and some protists. Plant cell walls are made of cellulose, fungi walls of chitin, while animal cells do not have a cell wall. This is why animal cells are more flexible and can form various shapes, whereas plant cells are rigid and maintain a fixed shape. Let us look at the DNA arrangements. In prokaryotic cells, DNA is usually circular and floats freely in the cytoplasm in a region called the nucleus. Let's look at DNA arrangement. Prokaryotic DNA is usually circular and floats freely in the cytoplasm in a region called the nucleus. They may also carry similar smaller circular DNA molecules called plasmids, which can confer special traits such as antibiotic resistance. Eukaryotic DNA is linear, packaged into chromosomes and enclosed within a nucleus. These organizations allow eukaryotic cells to manage much more genetic material efficiently, regulate gene expression, and coordinate complex processes like growth, differentiation, and reproduction. With organelles, prokaryotic cells lack membrane-bound organelles. Everything happens in one compartment. They have ribosomes for protein synthesis, but these are smaller, called the 70S ribosomes. While the eukaryotic cells have multiple organelles. Mitochondria produces energy in the form of ATP. Endoplasmic reticulum helps make proteins and lipids. Golgi apparatus packages ships proteins. Golgi apparatus packages and ships proteins. Lysosomes break down waste, while chloroplasts in plants convert sunlight into energy. In reproduction, prokaryotes reproduce through binary fission, a rapid process where one cell splits into two identical cells. This allows bacteria to multiply quickly in favorable conditions. Eukaryotic cells reproduce via mitosis for growth and repair, and meiosis for sexual reproduction. Mitosis has multiple stages: the prophase, metaphase, and the anaphase, including the telophase, ensuring precise division of DNA into daughter cells. A fascinating application is the antibiotics. Because bacterial cells have unique structures like peptidoglycan walls and 70S ribosomes, drugs can target this without harming human cells. This is why penicillin kills bacteria but does not affect our own eukaryotic cells. Now let's pause and think. Imagine designing a drug. Understanding the structural differences between prokaryotic cells and eukaryotic cells is absolutely critical. Without this knowledge, you could harm human cells while trying to kill bacteria. Next, we'll look at the functions of the structures we just described. The cell membrane. It controls what enters and exits the cell, maintaining homeostasis, nutrients, water, and ion passing, while waste products are removed. The cytoplasm is a jelly-like medium where chemical reactions occur. In prokaryotes, it carries out all life processes, while in eukaryotes, it supports organelles and helps in transport. Ribosomes are protein factories. Prokaryotes have smaller 70S ribosomes, while eukaryotes have larger 80S ribosomes. Proteins are essential for enzymes, structure, and signaling. The nucleus stores DNA, controls gene expression, and regulates cell activities. Mitochondria are powerhouses producing ATP. Cells like muscle cells have many mitochondria because they need a lot of energy. Chloroplasts capture sunlight for photosynthesis in plants and some protists. Even though prokaryotes have no organelles, they are still incredibly efficient. For example, bacteria can metabolize sugars, replicate DNA, and divide all in one compartment. Let's take a case study. The Messelson and Stahl experiment in 1958 demonstrated DNA replication in E. coli. Using isotopic labeling, they showed that DNA replication is semi-conservative. Each daughter DNA molecule has one strand and one new strand. This experiment confirmed how prokaryotic DNA replicates and was a key step in understanding genetics. Cells are not just theoretical, they impact our daily lives. In medicine, prokaryotic cells are the target of antibiotics, while eukaryotic cells are the focus of cancer research and gene therapy. For example, understanding the cell cycle in eukaryotes help design drugs that stop cancer cells from dividing. In biotechnology, E. coli is engineered to produce insulin for diabetes patients. Yeast, a eukaryote, is used in baking, brewing, and bioethanol production. In environmental science, bacteria help in nitrogen fixation, which enriches soil for agriculture. They also treat wastewater. Plant eukaryotic cells are being explored for biofuel production. Let us have this thought exercise. Imagine a world without eukaryotic cells. Humans, animals, and plants wouldn't exist. Now imagine a world without prokaryotic cells. No bacteria to fix nitrogen, no microbes to recycle nutrients. Both cell types are essential for life as we know it. Let us ask these questions. Can eukaryotic cells survive without mitochondria? Well, some energy can come from glycolysis in the cytoplasm, but generally, mitochondria are essential. Are viruses cells? No. Viruses cannot reproduce or metabolize on their own. They need a host cell. Do all eukaryotes have a cell wall? Only plants, fungi, and protists. Animal cells don't. Why don't antibiotics harm human cells? Because they target bacterial specific structures like the 70S ribosomes or cell wall, which humans do not have. In summary, prokaryotic cells are simple, lack a nucleus and organelles, and reproduce by binary fission, while the eukaryotic cells are complex, have a nucleus and organelles, and reproduce by mitosis or meiosis. Remember, structure drives function. Prokaryotes perform all tasks in a single compartment, while eukaryotes have specialized organelles for each function. Think of it like this. Prokaryotes are tiny studio apartments, while eukaryotes are houses with different rooms. Both work, but the house allows more complex activities. Thanks for watching. If you enjoyed this video, don't forget to like, subscribe, and click the bell for more notifications. Keep exploring the incredible world of cells and the microscopic engines of life.